ever looked at a really big old tree and just wondered, how does it get so incredibly wide?
Mhm.
Or, how can it live for literally hundreds, even thousands of years? It's not just about growing taller. Plants have this whole other secret life, growing outwards, building up that strength year after year. So today we're doing a deep dive into exactly how plants achieve that impressive girth. We're drawing our insights straight from a chapter in Raven Biology of Plants, the 8th edition. It's a real cornerstone
A
text.
classic, definitely.
Right. Our mission today is to unpack the fascinating processes of secondary growth in stems. Think of it as a shortcut to understanding the hidden mechanics that let trees and other woody plants build these incredibly robust, long-lasting structures.
Yeah, getting into the real nuts and bolts of it.
Exactly. We'll be exploring the two key players, these lateral meristems, the vascular cambium, and the cork cambium. They really orchestrate this whole thickening process.
And we'll see how this differs, between plants that really go for this thickening versus those that mostly just focus on getting longer.
Okay, so let's start with a foundation from the book, Plant Life Cycles. This really sets the scene for why secondary growth is, well, so important for certain plants.
Absolutely. Context is key.
First up, you've got annuals. These guys complete their entire life cycle, seed, growth, flowering, new seeds, all within a single growing season, sometimes just a few weeks.
Live fast, die young, basically.
Pretty much. Think about lots of weeds, wildflowers, many garden flowers, vegetables like corn or peas. Only the dormant seed makes it to the next season. Then you have the biennials. Their life cycle spans 2 growing seasons.
2 years, right?
Yeah. First year, it's all about vegetative growth roots, maybe a short stem, often a rosette of leaves close to the ground. They're storing up energy.
Building resources.
Exactly. Then, second season, boom, they flower, make fruits and seeds, and then they die off. And the source makes this interesting geographical note. In the southern hemisphere, they kind of act like winter annuals because of how the calendar falls.
Huh, that's neat. But neither annuals nor biennials really get woody, do they?
Generally not, no. They might have a little bit of secondary growth, but nothing substantial. They don't usually build up that thick, woody tissue. And that brings us to the perennials. These are the plants where the vegetative structures live on year after year.
The long haul plants.
Right. And we can sort of split them into two groups. You've got herbaceous perennials. Think of things like irises or hostas. They die back above ground during tough seasons, but survive underground with roots or maybe rhizomes, bulbs, tubers.
Waiting it out underground.
Yep. Then you have the woody perennials. These are the vines, shrubs, and trees we typically picture. They survive above ground. They just stop growing during unfavorable seasons.
And these can live for a really long time.
Oh, absolutely. Some take ages just to flower. Like the book mentions the horse chestnut might take about 25 years. And then there's this Puya Raimondi in the Andes. Apparently it can take around 150 years to flower.
Wow, 150 years. That's incredible patience.
Isn't it? And even evergreens, they shed and replace leaves, but not all at once like deciduous trees.
So what's really fascinating here, connecting it back, is how these life cycles directly link to the need for structural support.
Right?
You see, that ability to get really tall, really massive, like we see in so many perennials, it's tied directly to their capacity for the secondary growth.
That thickening process.
Exactly.
Yeah.
It provides the strength, the stability needed to support that size and weight and to live for so long. It's all about building a robust, long-term structure. And the main tissue doing this thickening is secondary xylem, which, we just call wood.
Okay, this is where it gets really interesting for me. Let's dive into that internal engine, the vascular cambium.
The master builder.
Totally. It's the unsung hero of plant girth. Our source, Raven, points out that unlike the apical meristems at the tips that make plants longer, the vascular cambium is a lateral meristem. Its cells are different too, highly vacuolated.
It's specialized for widthwise growth.
Precisely. And it has two main types of cells, two kinds of initials. First, there are the fusiform initials. These are vertically oriented, much longer than they are wide.
Okay, vertical.
Yeah. Think of them as forming the main pipeline system, the axial system, running up and down the stem. This is for the secondary xylem and phloem.
Got it. Yeah, the second type.
Those are the ray initials. These are oriented horizontally, sort of squarish or slightly elongated.
Horizontal, so running outwards.
Exactly. They form the vascular rays, which make up the radial system. Think of them like the spokes in a wheel, connecting things horizontally.
Okay, so you've got vertical pipes and horizontal connections being built at the same time. That makes sense for strength and transport in all directions.
It's a really elegant system, and how does it actually produce these tissues? through cell division, of course, but two specific kinds.
Okay.
The really crucial ones for adding new tissue are periclinal divisions. Imagine the cell plate forming parallel to the surface of the stem.
Parallel, okay.
If a new cell is made towards the inside of the cambium ring, it develops into a secondary xylem cell new wood. If it's made towards the outside, it becomes a secondary phloem cell new inner bark. This creates these continuous radial rows of cells you see in wood.
So it's constantly pushing wood inwards and phloem outwards. But wait, if the stem is getting wider, the cambium ring itself has to get bigger too, right? Otherwise, it would just split.
Excellent point. That's where the second type of division comes in. Anticlinal divisions.
Anticlinal.
Yeah, these divisions happen perpendicular to the surface. They essentially add new cambium initial cells into the ring itself, increasing its circumference.
Ah, so it's adding new builders to the crew as the construction site gets bigger.
Exactly. It's like adding new segments to keep a growing circle intact. So the cambium expands right along with the stem. The main products, again, are secondary xylem, wood, inside, and secondary phloem outside.
And those vascular rays you mentioned, the horizontal system, what's their specific job?
Super important. They're the pathways for moving food from the phloem across to the xylem and water from the xylem out to the phloem. Plus, they act as storage centers, starch, proteins, lipids, and they can even synthesize some secondary compounds.
So transport, storage, even some chemical work. Very versatile.
Definitely. Now, sometimes the term vascular cambium just means those initial cells, but often it refers more broadly to the cambial zone, which includes the initials and their immediate derivatives that haven't quite differentiated yet.
Okay, a zone of active division and early differentiation.
Right. And its activity isn't always constant. In places with distinct seasons, like temperate regions, the cambium usually goes dormant in winter.
Takes break.
Yeah. Then in spring, it reactivates. The cells take up water, they swell, they start dividing again. This reactivation actually makes the bark slip easily in spring if you've ever peeled a willow twig, that's why.
Ah, interesting. What triggers that reactivation?
It's hormonal. Specifically, the hormone auxin, produced by the newly developing shoots and buds way up top. As auxin moves down the stem, it basically tells the cambium, okay, time to wake up and grow.
A top-down signal. Makes sense. Coordinate growth with new leaves.
Exactly. Things are a bit different in some tropical regions where growth might be more continuous or respond to wet-dry cycles instead of temperature.
So this whole process raises a key question for me. With all this continuous addition of cells, how does the plant maintain its structural integrity? It seems like it could get messy.
That's where the organization comes in. Those continuous radial files of cells produced by the periclinal divisions create an incredibly strong integrated structure.
Right, the rows line up.
And the precision of having periclinal divisions for adding tissue and anticlinal divisions for expanding the cambium itself, it's remarkably controlled.
It really highlights the plant's dynamic response, adapting its growth engine based on internal signals like auxin and external conditions.
So, thinking about the stem we started with, the young primary stem, what does all this secondary growth actually do to it? How does it transform that initial structure?
Good question. Where does the cambium even come from initially in this stem?
Great question. It originates from two places within those vascular bundles you find in young stems. Part of it comes from the procambium undifferentiated cells located between the primary xylem and primary phloem.
Okay, within the bundles.
Right. And the other part arises from parenchyma cells in the interfascicular regions, the areas between the vascular bundles.
Connecting the dots, essentially.
Exactly. So you get fascicular cambium inside the bundles and interfascicular cambium between them. And critically, unlike in roots, these two connect up pretty quickly to form a complete continuous cylinder early on in the stem's development.
Ah, so it starts as a full ring right away in stems.
Generally, yes. And as the cylinder gets going, it produces secondary xylem and phloem, forming continuous cylinders of these tissues interwoven with those vascular rays extending radially outwards.
And the book notes, significantly more wood is produced than phloem each year.
Yes, much more secondary xylem than secondary phloem, which
Yeah.
makes sense, for structural support. Wood provides the bulk of the strength.
So what happens to the original primary tissues as this cylinder expands, like the primary phloem?
Well, they get pushed outwards. The primary phloem, being relatively delicate, mostly gets crushed and destroyed. Sometimes, if there were thick-walled primary phloem fibers, those might persist for a while, but the conducting cells are gone.
Sacrificed for the greater good of the expanding structure.
Pretty much. You can picture, like the book describes for an elderberry stem, starting with just small amounts of secondary growth, but by the end of the first year, there's way more secondary xylem than phloem. The book also mentions basswood stems as an example, showing 1, 2, and 3-year-old growth. And it points out how some vascular rays, called dilated rays, actually widen significantly towards the outside
Dilated rays, why would they widen?
to help accommodate the increasing circumference. As the stem gets thicker, the outer tissues need to stretch or expand somehow. These widening rays help manage that strain and maintain connections.
Clever. So the whole system, root and stem, is connected.
Absolutely. The vascular cambia and the secondary tissues of the root and stem are continuous. There's no abrupt transition region like you see between primary root and shoot structures. It's one integrated system.
Connecting this back, it really shows that growth isn't just layering stuff on, it's a dynamic reorganization.
Yeah.
The plant actively dismantles or modifies its earlier structures, like the primary phloem, to make way for the secondary body, prioritizing that long-term strength and transport capacity. It's a fundamental trade-off for becoming woody and long-lived.
Okay, so internally we have this massive expansion, but what about the outside? That original skin, the epidermis, it's delicate. It can't possibly stretch enough to cover a growing tree trunk.
Right, it's got to rupture eventually, so what takes its place?
That's where the next layer of defense comes in, the periderm. Think of it as the plant's replacement skin, but much tougher, like a sophisticated, multi-layered shield.
The outer armor.
Exactly. The periderm replaces the epidermis as the protective covering on stems and roots undergoing secondary growth. And it's actually made of 3 parts.
3 layers to this armor.
You got it. First, there's the cork cambium, also called the phellogen. This is another lateral meristem, like the vascular cambium, but its job is to produce the periderm.
Okay, the factory for the outer layer.
Yep. Then, produced outward from the cork cambium is the cork itself, technically called phellum. These are the cells that become heavily impregnated with suberin and wax.
So, Rand, that makes them waterproof, right?
Totally. Impermeable to water and gases.
Oh.
And because of that, these cork cells die once they're mature. They form the dead, protective outer layer.
So, waterproof and dead. Tough stuff. What's the third part?
The third part is formed inward from the cork cambium. It's called the phelloderm. This is actually a layer of living parenchyma cells, kind of like the cortex tissue.
Living cells on the inside. Interesting. So where does this first periderm usually form?
It typically shows up during the first year of growth. Most commonly, it arises from cortical cells lying just beneath the epidermis, but sometimes it can actually start in the epidermis itself or even deeper, maybe in the primary phloem.
Okay, so it replaces the epidermis early on. But wait, if the cork cells are impermeable, waterproof, and gasproof, how do the living tissues inside the stem, like the phloem and the cambium and the rays, get the oxygen they need for respiration and get rid of CO2?
Excellent question. That would be a major problem, sealing everything off. But the periderm has a built-in solution, structures called lenticels.
Lenticels, like little openings?
Exactly. They are specific regions within the periderm where the cells are arranged more loosely with lots of intercellular spaces. They act like pores or vents, allowing gases to diffuse between the atmosphere and the living tissues inside.
So breathing pores in the armor.
That's a great way to put it. You can often see them on the surface. They look like small raised dots or lines. Think about the little spots on apples or pears or lines on birch bark. Those are lenticels.
Oh, I've definitely seen those.
They start forming right along with the first periderm, often developing underneath the original stomata of the epidermis. And as the bark gets thicker and cracks on older stems, new lenticels keep forming in those fissures.
That's really elegant design. The suberin in the cork provides this amazing barrier, super protective, but the lenticels are this clever compromise, allowing essential gas exchange without, weakening the overall protection too much.
It really is. It's a sophisticated adaptation for life on land, protecting those vital inner tissues while still allowing them to breathe. Okay, so we've talked about periderm. Now let's clarify a term people often use, sometimes loosely, bark.
Right. What exactly is bark, technically speaking?
According to Raven, bark includes all the tissues located outside the vascular cambium.
Everything outside the vascular cambium. So that includes?
That includes the secondary phloem, plus any remaining primary tissues like the cortex and all the periderm layers.
Okay, so secondary phloem, cortex maybe, and periderms. Got it.
And the composition of bark changes as the stem ages. Very early on, it might just be primary tissues. After the first periderm forms, it includes that, plus secondary phloem, and maybe some trapped primary tissue.
And the vascular cambium keeps adding new secondary phloem every year.
It does, but remember, usually much less than it adds secondary xylem. And what happens to the older secondary phloem? As new periderms form deeper inside the stem, the older phloem cells, especially the softer ones, get crushed and cut off from supplies. Eventually they die and become part of the outer dead bark that gets sloughed off.
Ah, that explains why much less secondary phloem accumulates over time compared to the wood, the secondary xylem, which just keeps building up.
Exactly. So this leads to a distinction within the bark itself. Inner bark versus outer bark.
Okay, what's the difference?
The inner bark is the living part. It extends from the vascular cambium outwards to the innermost active cork cambium. This is where you find the functional conducting phloem, usually only the most recently formed layer that's actively transporting sugars.
The working phloem.
Right. The inner bark also includes older, non-conducting phloem layers where the sieve elements might no longer be functional, but the parenchyma cells are still alive, often used for storage.
And the outer bark.
The outer bark is everything outside that innermost cork cambium. It's composed entirely of dead tissues, all the old, accumulated periderm layers, and any dead cortical or phloem tissues trapped between them. This is the really tough, protective, non-living layer you typically touch on a tree.
So inner bark is alive and includes working phloem. Outer bark is dead and purely protective. That makes sense. It shows this dynamic layering, this history of protection being built up.
And how these new periderms form influences how the bark looks on the outside. If subsequent cork cambia arise in discontinuous overlapping patches.
Like scales.
Yeah, exactly. You get what's called scale bark, like you see on pine or pear trees.
Yeah.
If the new cork cambia form as more or less continuous cylinders,
Like rings.
right, you get ring bark, which is less common, seen in things like grapevines or honeysuckle. Many barks are kind of intermediate between the two.
Texture depends on how the new armor layers are laid down.
Precisely. And the book gives this fantastic commercial example, the cork oak, Quercus suber.
Ah, where bottle corks come from.
Exactly. The first periderm it makes, the virgin cork, isn't very useful commercially. But after the tree is about 20 years old, this original layer is stripped off.
Doesn't that hurt a tree?
It stimulates a new cork cambium to form deeper in the living tissues, and this new cambium produces really thick, high-quality cork. This layer can then be harvested carefully about every 10 years or so for maybe 150 years.
Wow, that's sustainable harvesting.
It really is. And if you look at a wine cork, those little spots and streaks you see, those are the lenticels, the breathing pores we talked about, preserved in the harvested cork.
That's amazing. A perfect example of harnessing a plant's natural protective growth built up through all these secondary processes.
Okay, let's shift focus now to the inside, to what makes up the bulk of the stem in woody plants. Wood itself, the secondary xylem.
Arguably the most important plant tissue for human civilization, right?
Yeah.
Shelter, fire, tools, paper. It's everywhere.
Absolutely indispensable. Now, botanically, woods are often classified into two broad groups.
Hardwoods and softwoods.
Right. Hardwoods are generally the woods from angiosperms specifically, the magnolids and eudicots like oak, maple, balsa. And softwoods are the woods from conifers like pine, fir, spruce.
But those names can be misleading, can't they?
Definitely. It's important to remember that hardwood and softwood don't reliably indicate the actual physical hardness or density. Balsa, for example, is technically a hardwood, but it's incredibly soft and light. And some softwoods, like certain pines, can be quite hard. It's really a botanical classification based on the type of tree.
Got it. So, structure first. What's characteristic about conifer wood, the softwoods?
Simplicity and uniformity are key. The most striking feature. Conifer wood lacks vessels.
No vessels, those wide pipes we see in angiosperms.
Nope. Instead, their axial system, the main vertical component, is made up almost entirely of one cell type, tracheids. These are long, tapering cells that handle both water transport and structural support.
Dual function cells.
Right. There's usually very little axial parenchyma, the storage cells, except sometimes around resin ducts, which you find in pines, for instance.
Resin ducts for protection.
Probably. They're channels lined with cells that secrete resin, likely as a defense against fungi or insects. A key feature of conifer tracheids are their bordered pits. These are specialized pit pairs, usually large and circular, found mostly on the radial walls where the tapered ends of tracheids overlap.
And these pits have that special structure, the torus.
Exactly. The pit membrane has a thickened central part called the torus. It can act like a valve. If one tracheid gets blocked by an air bubble, an embolism, the pressure difference can push the torus against the pit aperture, sealing it off.
Clever. Preventing the air bubble from spreading to the next tracheid. A safety mechanism.
A crucial one, especially for trees facing freezing conditions. If you look at conifer wood in different sections, you see this structure clearly. Transverse section shows angular tracheids in rows with narrow rays. Radial section shows the rays as sheets running perpendicular to the tracheids. Tangential section cuts through rays, revealing their height and width. In pine, they're mostly just one cell wide.
Relatively simple, uniform structure compared to hardwoods.
Much more so. Angiosperm wood, or hardwood, is generally far more complex and varied.
Why the complexity?
Mainly because it contains a greater variety of cell types. The defining feature, with very few exceptions, is the presence of vessels.
The wide pipes for water transport?
Right. Vessel elements join end to end to form these long, efficient pipes. But angiosperm wood also has tracheids, plus specialized fibers for support, and typically more abundant axial parenchyma cells for storage.
More specialized roles for different cells.
Exactly, and their rays are also more diverse. They can range from just one cell wide, like in conifers, to many, many cells wide. Think of oak wood. Its rays are huge, easily visible, sometimes up to 30 cells wide and hundreds high. Hardwood rays make up a larger percentage of the wood volume too, maybe around 17% compared to about 8% in conifers.
So overall, angiosperm wood is structurally more complex, more specialized cell types, larger rays, maybe less orderly sometimes.
Yeah, the book mentions it can appear less orderly than conifer wood, partly due to the variation in vessel size and arrangement and how fibers elongate. Now, one of the most familiar features of wood, especially from temperate regions, are growth rings, or annual rings.
The things we count to tell a tree's age.
Typically, yes. They result from that periodic activity of the vascular cambium we discussed, active growth in spring and summer, dormancy in winter. One layer of growth usually equals 1 year, forming an annual ring.
Usually. Can it be
Yeah.
inaccurate?
It can. Sometimes abrupt environmental changes during the growing season, like a sudden drought followed by rain, can cause the tree to form what looks like an extra ring, a false annual ring. And in some tropical areas with continuous growth, trees might not form distinct rings at all.
So counting rings isn't always foolproof for age.
Not always, but it's often a very good indicator. And the width of the rings is incredibly informative. It acts as an environmental index. Wide rings generally mean good growing conditions, enough rain, favorable temperatures. Narrow rings mean stress, drought, cold, maybe insect infestation.
Like a historical record written in the wood.
Exactly. The book highlights the bristlecone pine as an amazing example. Some living ones are nearly 5,000 years old, and dead wood samples go back over 8,000 years. Scientists called dendrochronologists study these rings as incredibly sensitive records of past rainfall and temperature.
Wow. So what actually makes the ring visible? What's the structural difference within one ring?
It's all about density changes between the wood formed early in the growing season and that formed later. The early wood, or spring wood, forms when growth starts rapidly. It typically has wider cells, vessels or tracheids, with thinner walls, making it less dense and better for rapid water transport.
Okay. Wider pipes, less dense.
Then, as the season progresses, the late wood or summer wood is formed. It usually has narrower cells with much thicker walls, making it denser and stronger, providing more support.
Narrower cells, denser wood.
Right. And the transition from the dense late wood of 1 year to the much less dense early wood of the next spring is usually quite abrupt. That sharp contrast is what makes the annual ring boundary so distinct.
That makes perfect sense. Early wood for flow, late wood for strength.
And based on how the vessels or pores are distributed, we can classify angiosperm woods further. In ring porous woods, like oak or ash, the really large diameter vessels are concentrated mainly in the early wood of each ring.
Big pipes at the start of the season.
Yeah, it allows for very rapid water transport, mostly happening in the outermost growth ring. In contrast, diffuse porous woods, like maple or birch or the tulip tree mentioned, have vessels or pores that are more uniform in size and distribution throughout the entire growth ring.
Different strategies for water transport across the season.
Exactly. Now, another key distinction within the wood of an older tree is between sapwood and heartwood.
I've heard those terms. Sapwood is the outer part.
Correct. As a tree ages and the trunk gets wider, the older inner wood eventually stops functioning in water transport and storage. The sapwood is the physiologically active, usually lighter colored outer region of the wood. It contains living parenchyma cells, stored food reserves, and it's where water conduction primarily occurs, although maybe not all of it is conducting at any given time.
And the heartwood.
The heartwood is the inner core of often darker, non-living wood. It forms as the tree ages and withdraws nitrogen and other resources from the oldest sapwood layers. It then deposits various secondary metabolites, things like oils, gums, resins, tannins, into these cells.
Waste products or defensive chemicals?
Probably a bit of both. These substances often make the heartwood darker and significantly more resistant to decay in insects, but they also kill the remaining living parenchyma cells. So heartwood is essentially dead, non-conducting tissue, primarily providing structural support. The relative amount of sapwood versus heartwood varies hugely between species.
So sapwood, alive and working, heartwood dead and durable. Sometimes you see blockages in vessels too, right? Tyloses?
Yes, tyloses are fascinating. They're balloon-like outgrowths from adjacent living parenchyma cells that protrude through the pits into vessel elements. They can effectively plug up the vessel.
Why would the tree do that?
It's often a response to wounding or infection, basically walling off the damaged or infected vessel to prevent the spread of pathogens or air. They also commonly form during the transition from sapwood to heartwood.
Another protective mechanism built into the wood structure.
Indeed. And speaking of structure adapting, there's reaction wood. This is wood formed specifically in response to mechanical stress, particularly gravity, in leaning trunks or branches.
The tree trying to correct its posture.
Essentially, yes. In conifers, it's called compression wood. It forms on the underside of the leaning stem or branch. The cambium becomes more active there, producing wider growth rings with tracheids that are heavily lignified. This denser wood physically pushes the stem upwards.
Pushing from below.
Right. In angiosperms, it's called tension wood, and it forms on the upper side of the lean. Here, the extra growth effectively pulls the stem upright. Tension wood is characterized by special gelatinous fibers, which have an inner wall layer rich in cellulose and lacking lignin.
Pulling from above. Different strategies, same goal. Does this reaction would cause problems?
It can in timber. Both compression and tension wood tend to shrink and swell unevenly when drying, which can cause warping or twisting in lumber. Finally, the book touches on wood density and specific gravity. Density is simply weight per unit volume, and it's probably the most important predictor of wood strength, hardness, how well it holds nails, even its quality as fuel.
Denser means stronger, generally.
Right. Generally, yes, though denser woods also tend to shrink and swell more with moisture changes. Specific gravity is related. It's the ratio of the wood's oven-dry weight to the weight of an equal volume of water. The actual wood substance itself has a specific gravity of about 1.5, regardless of species. So differences in the specific gravity of different woods depend on how much solid wall substance there is compared to empty space, the cell lumens.
So thick-walled cells with narrow openings make for denser, higher specific gravity wood.
Exactly, like wood rich in thick-walled fibers.
Looking at all this complexity, the simple tracheids and conifers versus the complex vessels and fibers and angiosperms, the different ray structures, ring porous versus diffuse porous reaction wood, it makes you wonder, how does a plant decide which wood structure is optimal for its particular life and environment?
That's the multi-million-dollar evolutionary question, isn't it? It's not really a conscious decision, of course. It's the result of millions of years of natural selection, constantly fine-tuning that balance between structural support, efficient water transport, cost of building the tissue, and resilience against drought, freezing, pathogens, and physical stress. Each strategy represents a different evolutionary solution to those challenges.
Wow, okay, what a journey through the inside of a stem.
Yeah.
We really peeled back the layers today, quite literally.
We did, from life cycles to bark to the heart of the wood.
Yeah, we started by seeing how the different life cycles, annual, biennial, perennial, set the stage for whether a plant needs substantial secondary growth.
Right, perennials needing that long-term structure.
Then we dove into the powerhouse, the vascular cambium, that incredible engine-producing wood, secondary xylem, inwards and inner bark, secondary phloem, outwards, allowing the stem to thicken.
Driven by those specific paraclinal and anticlinal divisions. Exactly, and we saw how that impacts the primary body, pushing aside old tissues. Then we looked outwards at the protective periderm formed by the cork cambium, with its waterproof cork cells and those vital breathing pores, the lenticels.
The armor plating with vents.
Perfect description. We clarified what bark actually encompasses everything outside the vascular cambium, distinguishing between the living inner bark and the dead outer bark.
And why less phloem accumulates than xylem.
Right. And finally, we explored the amazing world of wood itself, secondary xylem. We saw the differences between simpler conifer wood and more complex angiosperm wood.
Hardwoods versus softwoods, vessels versus tracheids.
Understanding growth rings not just for age, but as environmental records, distinguishing functional sapwood from durable heartwood, and even looking at how trees respond to gravity with reaction wood.
It's incredibly intricate.
It truly is. It's such a testament to the sophistication of plant biology that these processes allow a tiny little seedling to potentially become this colossal, long-lived organism standing strong for centuries.
A silent, powerful engine of growth hidden inside.
Yeah. So maybe the next time you look at a tree, you won't just see the leaves or the flowers. Maybe you'll think about that hidden engine beneath the bark, the cambium, tirelessly adding new rings, recording history, building strength layer by layer. It really makes you wonder, what other hidden wonders, what other intricate processes are constantly shaping the world around us in ways we rarely stop to appreciate?